So welcome everybody. Today I'm going to talk about modeling brain behavior dynamics in freely moving subjects. And I will try to give you a translational and ontogenic perspective on that. So as a first thing, I would like to focus briefly on discussing a little bit what defines or how have been classical lab models conducted so far, also classical paradigms therefore, and what defines a freely moving behavior. So when modeling brain behavior dynamics in classical lab settings, the paradigms are designed in a way that you can isolate the specific cognitive process that you're interested in. And that also means that the paradigms are often designed in a way that you have a discrete time that can turn into repeated trials because you have a very defined stimulus and a very specific response, which is a single behavior variable of interest that you are interested in and you want to study. And that the paradigm repeats and repeats, and therefore you get many, many trials. So the event that you were interested in, the response that you were interested in, will be expressed many times. You can time average, and then you can analyze the signals. And subjects here are usually seated and also highly constrained, so they even are often putting their chin on a chin rest or have a head fixation, and so on and so forth. So movement, if it exists at all, is considered to be completely noise, and it's really tried to keep at a minimum. And these choices are very deliberate, and have been also extremely successful in the past. They have given us very high control over the paradigms, and therefore we got to know a lot of scientific findings that we would have not known otherwise. And it does help because it constrains the data, and therefore these constraints help to get us clean data. And we can, with that, then align neural signals across trials. We can average the responses. We can build models with relatively few degrees of freedom. So as you can say, from a neural data science perspective, this is already seems to be an ideal regime. And in this setting, I mentioned it in a side note, but I want to focus on this now. Movement is considered noise. And if subjects shift posture or move in unexpected ways, we often try to exclude those tries or average this response out in some way. We just don't want to have movement in the data at all. And the underlying assumption when we try to model the data or analyze the data from these classical lab settings is that cognition leads motor execution. So it's a one-way link. So it's really kind of implying that whatever happens in the brain happens without any movements. You will do all the deliberation, you will do all the cognitive processes that you're interested in, and then only you will say, okay, now motor, um, my motor system, please start executing what I just decided on doing. And this assumption is deeply embedded how we analyze the data, as I mentioned already, even though it's maybe not said so explicitly, but if you think about it, I hope that makes sense. But if you look at real life, how does it look like? So we have more than just simple arm reaches or saccades, which is studied often in classical paradigms, be it in humans or be it in rhesus macaques. We can climb, we can move our whole body. And here you can also see examples. We are also social species, obviously, that also plays an important role. But also often we are navigating and deciding as we go. So it's actually not really that we are deciding or thinking about things while having no movement at all, and only then it translates into movement. But actually, it seems to be that movement and cognition is temporarily overlapping, actually. And that's what leads me to the freely moving aspect of my studies that I am now very interested in and the direction I'm going with my research. So in freely moving, how I would define is that the agent is not sitting still and just waiting for a stimulus, but the agent is continuously sampling the environment and is moving, is orienting, it's by doing all of this, updating its internal state continuously. And there are no clear trial boundaries, so you don't have a start trial, end trial. It's all continuous movements, and you don't also often have necessarily singled out response variable that would explain the behavior or the cognitive process you would be interested in. And therefore, you also do not get necessarily obvious moment for decision points because all cues were available, as it would be the case in the classical settings where you're sitting, you receive all the information that you need, then there is a delay period, which just means that you now have time to process all those cues that you just received, make a decision, make up your mind, and then comes the go cue, which is, okay, now you can start acting upon that. You don't have this in freely moving because it's not that you're having a clear, that was my cue, that was my cue, but maybe a certain environmental object was a cue for one person and another one would be for another person. And this already changes quite the nature of the modeling problem, as you can imagine. So the key contrast in freely moving and real life is the cognition is not something that happens first and then gets only expressed through movement. So it's not this one-way link. But in my opinion, and also some other, obviously, it's not something that I came up with, have shown some intuitions about this and also some results that show that cognition and movements are actually highly interconnected, and perception, decision making, and actions are intertwined in continuous time. And that leads me to the concept of embodiment, and that's the second section I would like to talk about. So, embodiment, you can think about that as a useful framework of thinking. What embodiment is trying to, oops, and here we go. What embodiment is trying to say is that the emphasis is not that cognition is something that happens independently from the movement in the brain, but body plays an active role in shaping how information is acquired in the first place, how it's processed, how it's acted upon. And that leads me to this brain body loop. So, an embodied agent will have, if you imagine now that you have the neural activity that drives an action, so that would be the classical model. But then what happens is that that action will change the agent's position or posture, or orientation, or maybe also just something in the environment. And this again will reshape what type of sensory inputs the agent can receive in the first place. And this in turn will influence again the neural activity. So as you can see, it seems for me more plausible that we have a closed loop between brain, body, and environment. And it's not a one-way link from cognition to physical exploration, but actually the physical exploration also has a direct influence on the cognition. So in other words, how I like to think about it is where we go determines what we can learn and what we can do. And this is an important second half of this loop that could not be studied until now or has been neglected a little bit. And why this matters for modeling. So from the modeling perspective, it does has quite some consequences. So many variables are treated so far as independent in classical tasks, but in freely moving, they do become highly coupled. For example, you have gaze directions that would affect what information is sampled, and that would be temporarily completely separated in classical tasks because you would have some direction, then you would get an information, then you can move again your gaze, and then only you have to stop and only the information will come in. But in freely moving, its gaze direction affects what information is sampled, which then affects your decision, which affects your movement, and which affects the gaze again. So you again get this closed loop between physical exploration and cognitive exploration, or motor versus cognition. And these are not just side effects, but they are actually also true mechanisms. So what we also have, another distinction here is that in classical models, I mentioned it already, we have often a temporal separation between perception, cognition, and action. But in freely moving paradigms, and also especially in real life, that just challenges the idea of this clean processing stages because we have these temporal overlaps. In embodied agents, these distinctions are blurred because, as you can see here now, or as I tried to show, in classical paradigms, you would often have the cue, then comes a separate phase where you can deliberate, then comes a different phase where you can decide, and then say, okay, now you can move. In real life, cue, deliberation, and decision are often overlapping with ongoing actions, even. So, when does perception end and decision making begin? Or when does cognition stop and action planning start? In many cases, these questions are no longer having clear answers. And that now would lead me to the third part of this talk today, which is the modeling goals in neural data science. So I'm sure that you have heard a lot about this now, and you also have your own thoughts. I just want to present how I think about this. It might not be right, it might not be wrong. It's just to give you some perspective. So for me, there are kind of three overarching goals why we model neural data. It is either I want to predict, I want to infer, or I want to explain the link between cognition and motor. So if we focus on the prediction, in classical tasks, usually we often predict the next choice or the neural response or the next trial. And the targets are extremely well defined, and the timescales are very fixed. We just talked about this. In 3D moving, predictions become way more ambiguous because we cannot even define sometimes exactly what are we predicting. Is it the next step? Is it the next gaze shift? Is it a decision? Is it a change in posture? Which of these many factors is driving in the end the neural activity that we are seeing? And these variables do unfold on different timescales and influence each other, and therefore predictions become a multi-scale problem. Now focusing on inference, which for me is estimating the latent states, means in many models we infer beliefs, values, or intentions that were underlying cognitive processes that the subject was going through. In freely moving, the notion of state itself unfortunately becomes unclear. Because is the relevant state now cognitive, motor, spatial, social, or a combination of all of these, or maybe yet another fifth thing? We are not completely sure, or we cannot be always completely sure because it just is so many things going on at the same time and overlapping in time. And we never or almost never observe the full state of the system. So we have to infer based on snippets that we see. Thirdly, explanation. So which variables matters and when. In 3D moving, number of potentially relevant variables becomes significantly higher because, as I said, it's not just this one cue or these two cues that I showed you, but my environment or my freely moving task where it might be just how the light is going into the room, and therefore I want to avoid one corner and go to the other corner, which might not have to do anything with, oh, I wanted to go to the red square versus blue square, where you can be very sure in classical paradigms. Additionally, also body orientation, head direction, gaze, proximity, recent visitation history, social context, and many more matter, but not all at all times. But at different times, different factors will have different weights. So explanation inherently becomes context dependent and also time dependent. And this is also where our models break. So up to now, I've just shown you freely moving behavior is challenging and violates many assumptions that underlie classical neural data analysis. Okay, but I would like to get more specific and where do things actually really break? And here I want to focus on a typical analysis pipeline of a neural data scientist because I think that's a good example because pipelines often also encode some assumptions. And now we can go through it step by step to understand which assumptions are still true and freely moving, which are not. So a typical neural data science pipeline could look like, or often have these steps in there, which is first you record the neural and behavioral data. After this, you pre-process and clean the data, and then you choose a representation. And after that, you would fit a model on this representation, interpret the results, and then you hopefully have some conclusions. So the structure is very familiar, but it's like something that neural data scientists do all the time. And in freely moving behavior, though, every single step of this pipeline is challenged, I would say. So let's focus on the data acquisition step. So the first step. In classical setups, you work very hard to minimize movements. You have head fixations, especially if you're working with animals. You have limited workspace, you have constrained postures, and these are all by design choices, and they are meant to stabilize the signal. When in freely moving, when agents start moving, this stability kind of disappears because movement creates motion artifacts. For example, if you're recording them with EEG systems in freely moving humans, you get signal dropouts, you get occlusion. If you're doing computer vision for behavioral tracking, you get alignment problems across modalities. So these issues are not random, they are structured and often though also correlating with behavior. So often all my movement artifact might correlate if you did not design it in a very smart way with exactly the moment of interest where cognition should have been also very important. And then you get this very strong correlation and then what to do about that. And variability is already no longer independent of the cognitive process we want to study, but it cannot also be treated as a nuisance noise. So it's not just, it's fine, it's not that much, but actually it can be quite a bit. So that and already like all the hardware challenges, you get your data, let's assume, we go to the next step, pre-processing and cleaning. Many pipelines do rely on the assumption what a clean signal should look like based on these classical, very isolated events. Freely moving behavior shows higher variance, which does not mean, though, that it's low quality. And sometimes that high variance might actually be even an indicator of something very interesting. For example, you have changes in neurosignals that coincide with posture shift or head movements. And now, based on the classical data, if you would apply those pipelines, it would dismiss all those moments as artifacts, even though they are actually highly coupled to information sampling and decision making. So it might be that the participant is just every time shifting the posture to have a very clear clue of I am now sampling information, but then you might disregard it based on classical setups and the assumptions underlying there, and then you would just get rid of the interesting signals that you actually wanted to analyze or the time periods that you wanted to analyze. So preprocessing itself becomes an interpretive step when you're dealing with freely moving data. And here, actually, I want to tell you a short story. So you should also, or you could check out if you're interested in this EEG Many Steps project. So they are a consortium of several labs that are doing freely moving EEG, which is now up and coming more and more. And they evaluated around 100 labs worldwide and saw that actually none of this lab is using a same or similar pipeline as other labs. So each of those around 100 labs are using almost 100 different pre-processing pipelines. And now with this Many Steps initiative, they want to collect all those pipelines, they want to collect data across labs, and then really systematically show what type of differences on which completely different results you're getting if you're not doing the pre-processing in a specific way or in the other way. So that's going to be a very interesting project. And that just shows you the problematic right now that we have. Moving on to the next part, now we cleaned our data, representation of behavior. In classical tasks, behavior is often represented by a small number of very discrete variables, such as choice, reaction times, or movement onset. In free moving behavior, this becomes very high dimensional, and we have to decide whether to repress posture or trajectory or velocity or gaze or social context, and many, many more, and also at what level of abstraction. So there is not really, at least not currently, a way of saying this is a complete neutral choice, but it really depends highly on what were you after, what was your research question, and how did you design your paradigm. And every representation will highlight certain aspects of behaviors and will hide others. So therefore, it's really important to select smartly the right ones. So that shows you what you have to do here already is representation itself becomes a modeling decision. Coming next to the modeling assumptions, many classical assumptions start to fail here. The latest, which is that modeling assumptions are, because we have modeling assumptions that are not correct for freely moving paradigms. So example, just to name one, is independence. Behavior variables are highly correlated sometimes in freely moving paradigms. So body orientation will affect gaze. Gaze will affect movement. Movement will affect sensory input. So you can see it's a chain of correlations almost. And it's highly correlated and highly interlinked. Where in classical setups, body orientation would not play a role. Gaze would be very isolated from movement, and movement would be very isolated from your sensory input, besides, of course, your sensory feedback that you have from your arm or gaze movement. So these correlations are now not incidental, but they are actually mechanisms, and often coinciding again with features that you are interested in. So it becomes a bit difficult to understand which to exclude, which to clean up, and which to keep. And then the last bit, the interpretation. Interpretation asks which variable will matter and what neural activity is representing these variables. And in freely moving, we can have multiple variables that are co-varying, as I've already mentioned now many times. And therefore, attributing neurosignals to one specific cue becomes very ambiguous. So is a neurosignal now related to a decision, as we were after, for example, in a research question? Was it just movement adjustment? Was it just a change in posture? Or was it just a shift in attention? And I'm sure there's a longer list that we could continue here. So I hope you can see the problem or the challenge behind this. Our models are not even always equipped at the moment to tell these apart. I now want to briefly come up to noise versus structure. So at this point, probably it is very tempting to say freely moving is just super noisy and is actually not worth studying. But I think that would be a very misleading framework because what often is called noise is actually structure that exists outside the scope of our current model. And one could think about this in this way. So really moving behavior reveals dimensions of variations that we have historically ignored or we could just not study. For example, posture as a latent variable. Classical setups, there is one posture and you maintain this. You could have not even think about that as a variable. Coupling between decision and movement preparation. Active sensing policy, so the neural control of sampling during movement. So as you can see, it opens up, if you think about it in the right way, yes, it's a big challenge, but it opens us so many super interesting questions that were not addressed so far. Not because they were unimportant, but because it's just super hard to measure. So until a long time, the technology was just not there. Recording freely moving single unit activity in monkeys or mice is something that came up just very recently, just a few years ago. Or mobile EEGs were just not that good back then. And now it's getting there and it's improving. And also because it's just hard to model, as I've just shown you. And why does this matter? It matters because it does affect prediction accuracy and interpretation explanation. So if our models would systematically ignore certain dimensions of our behavior that are there in real life, we are risking misattributing neural signals to wrong causes. And I'm not saying that classical paradigms now are not good to explain our world. I think we have gone like miles of these type of studies to really understand and have very impactful research done with these controls, and they are very important. But what I would like to advertise here is that while keeping on doing so, in parallel, we should start opening up to use the latest technologies that are out there and start using it to try to understand step by step closer to real life scenarios. So the takeaway for me would be is so it's not that freely moving behavior is unmanageable. The takeaway for me is that our current data science pipelines are just not optimized for it, and they are just optimized for a different regime, which is the classical paradigms. And freely moving forces us now to confront assumptions that were previously just, like I call it invisible, but basically just taking as granted. But now we have to rethink those assumptions, and I think that's very exciting. And this also brings us a step closer to real life. So now, after having given you this small introduction, I would like to also focus briefly on wild translational and ontogenetic approach. So for me here, it's really just about giving you some insights, how I think about things. Um, of course, that doesn't mean that that's right or that's wrong, but just like how I think about it and where I come from. So I think that humans are a great model to study, and obviously, for obvious reasons, we are very interested in understanding the human behavior and human cognition. But the human model is also limited because freely moving human does show us rich detail, but and we can track movement, gaze, posture, and interaction with the environment, but we do not have access to single neuron activity or population level neural dynamics in humans. But this temporal and spatial resolution that you would get with single neuron activity is needed to disambiguate competing mechanistic explanations. And as a result, many models of human behavior do remain a little bit under-accessible. Therefore, that's where I come from. I've worked with rhesus macaques for many years. And here, my claim is not that rhesus macaques behave like humans in every aspect, and therefore it's a great model, but more that the goal is to identify also mechanistic principles of brain body interactions that are conserved across primates, so across rhesus macaques and also humans. And we can study this now with macaques, but we also can now with macaques get access to single neurons, which is fantastic. So if you want to understand how decisions unfold over time, or how exploration is shaped by posture, gaze, context, and many more, we need a system where we can observe both behavior and neural dynamics in detail. And that's why I believe that working with rhesus macaques is still important. But why ontogenetic now? So ontogeny is, for me, like a fast evolution. So what do I mean with that? I do not study only adult humans, but I also study children. I study them or I'm starting to study them with very young ages, starting 3, 4 years until older, and you can do even younger. Why? Because I think it's super cool, it's super exciting, because if you look at something across development, you're seeing basically an approximation of a fast evolution. And over development, what is happening is the body is changing, the motor repertoire is expanding, the sensory systems are maturing, and cognitive strategies are reorganizing. And now you can study all of that. And the cool part is, if you do it in a smart way, you can now keep the environment very, very similar. And just the agent's interactions are changing dramatically just because of the different stages of development. And this makes development a really powerful test case for brain body models, in my opinion. So, what development let us test is by studying behavioral neural dynamics across development, we can ask, for example, which aspects of brain behavior coupling are stable across age? Which change as the body and sensory motor capabilities change? And which models can adapt to these changes without being respecified? And now linking species and ontogeny, if principle generalizes across development across species, I believe we have a much stronger evidence that it reflects also really a fundamental aspect of brain-body interactions, which in the end of the day, I'm very interested in. Conversely, one can also argue if a model will fail in development, it tells us something very important about its limitations, and it tells us also what type of mechanisms might not be the same across development. And that's why I work with rhesus macaques, because they allow the study of freely moving behavior with mechanistic neural axes. And ontogenetic approach, because development provides a natural stress test for generalization. And together, I think these approaches help us to move from descriptive models of behavior towards mechanistic models of brain-body interaction that can plausibly translate to humans. And now in the last bit, I would like to focus a little bit on some exemplary case studies from my research. So I am very much interested in the link between brain, body, and environment. In the first example case, or example research, I just want to show you focusing body-environment link. So what was done here is that kids that were like 3 years old were shown 4 different objects, and 2 of them now were either familiar or 2 of them were novel. So in this specific example, you can say that the wagon and the iguana are the novel object, so the child is not familiar with it yet, and the bear and the car is the familiar object. And what we were interested in is to see, does familiarity and novelty, which are 2 essential drivers of curiosity, and side note, curiosity is for me the intrinsic motivation of sampling information for your own intrinsic reward. And familiarity and novelty, so novelty is supposed to be a very hard driver for that. And we just wanted to see what happens when you're interacting with the environment with if you start analyzing the hand touches or hand interactions, which is always now called touch, and the gazes. Do we see some differences between the familiar, so the blue one, and the novel objects? And if you really look at this, at the gazes, sorry, the headings are missing, but this is the touch and this is the gaze times, doesn't seem that there's a big difference. And that is kind of contradictory to some assumptions that they had in the past, but those studies were eye tracking data studies, or like passive studies where kids are just watching specific things. And then they concluded that, listen, like the babies engage with novel objects way longer than with familiar objects. So then we asked ourselves, is maybe not the outcome itself the key when we move these paradigms to real life and to a free play setting, but maybe the pathway of how we get to interactions? And here now I jumped a little bit. So what I mean with that is we defined that an interaction with the environment can be either just by head gazes, just by touches, or by gaze and touch coupled. And we did find that gaze and touch coupled is usually the common case. And we wanted to understand, how do we get there? In theory, my naive assumption was I will look at something and then I will start touching it as well at the same time. So I basically end up in the pathway of gaze leading to both, as I will call it now. We did this analysis here on the left side for the familiar cases and here on the right side on the novel cases. And what's super interesting actually is that it seems that the pathways are reversed. So what happens is that in the familiar case, we get many cases. I'm very sorry because here now the arrow is missing, but you just have to trust me. So here was an arrow in blue. And what happens is that with familiar objects, kids are often actually just touching and then ending up in coordination. Whereas in the novel objects, even if they were touching it only, which happens already very rarely, they end up stopping to touch, just looking, and then only start interacting with it. And the interesting part here is that if you look at these results across experience, so across the time they have been interacting with the object, this pathway starts becoming more and more like the familiar pathway. So our conclusion here for now is that familiarization or learning might actually be reflected in pathways towards multisensory interactions rather than in absolute outcomes as it has been assumed until now in current literature. But those paradigms were often, as I said, screen-based and constrained and not in a free play setting. But all of this is hand-coded or was hand-coded data back then. And now we are working towards automizing these predictions. And while touches work quite okay using like computer vision, for example, also simple bounding box tracking can provide you those results, gazes are still a bit of a challenge. So here we try to combine key point tracking with Gazelle. And the main problem here is so often it works well, but there are failure cases. And that is because we're trying to solve with a 2D model, in a 2D image, something that is a 3D problem. And therefore, and most importantly, also the model assumptions of Gazelle that was trained and assumed on adults do not hold true for babies often. So one of Gazelle's key assumptions is when there is something very salient happening in the screen, so for example, something is moving, then the gaze will be biased towards that. Babies ignore it. But babies are working on or playing with their toy. The parent can wiggle around another toy. They are not gonna necessarily focus on the wiggling toy. They will just continue with their stuff. In monkeys, I've told you that I've also worked with monkeys. We implemented a different approach. So we have used key point data. Here you see two monkeys where we reconstructed the whole full body behaviors, and then the yellow cone were the different gazes. But here our problem right now is that we have now many free parameters. So the angle through which how much the cone should be tilted, the cone size, etc. What is the truth? How will we know where they were looking at for real? And we are also interested in, once we should have a nice approximation that we feel confident enough, what do the single neural correlates of head gazes look like in freely moving macaques? Because in classical setups, it has been shown, if you look to the left or if you look to the right, that is like explicitly shown in the neural data, in the premotor cortex, then you can really decode it. But is this true in freely moving? We are not sure, we don't know. So this already shows you some open questions. But next I want to really focus now on the three-way link between body, brain, and environment. And here I want to also show you some stuff that is already working, just to like keep us going and show you that it's not just noise and it's not just frustrating. So what we have shown and found out so far is that are the decision signals during free but repeated movements in macaques. So here on the left now you will see a video where the rhesus macaques will go across the room, and here the screens will show either red or low or blue. The red always indicates a high reward, the blue always low reward. So they will get a reward anyways, but they can maximize the reward by going to the high rewards thing. And what the monkey has to do is has to start walking, and mid-movement at a certain point, the cues will arrive and it has to decide and move. And now, based on computer vision, we can extract the nice trajectories whenever they decide to go to the left or to the right. Based on these trajectories, we are able to extract time points that we think are decision time points, and then we can align the neural data to it. So very bringing freely moving towards the more classical analysis regime. And here I'm not going to go in detail, but the important thing that you need to know here is, so this is the wrist velocity of the monkey, so it's oscillatory, which makes sense. So they step, then the other paw, then they step, then the other paw, and so on. And here you see the low dimensional representation. So this is a PCA of the motor cortex of this monkey. And you can see the circular movements, which have been shown also from classical paradigms. And we do not see any left and right distinction. What is super cool though, in the premotor cortex that is supposed to be also part of decision making, we see a very clear split for right and left choices. So what we can say already here, I'm not showing you the full-fledged study, but already here, I hope I can convince you that we can successfully extract decision time point and then the decision signals, so decision neurosignals from the brain during ongoing movement. So it's not just noise, because when we started the study, people were convinced we are not going to see decision signals. It's all going to be motion artifact, movement. There's nothing to take out of it. But we can, and that's what we're showing. And I think that's already very fantastic. And I hope I can excite you with that. One thing to consider here is we get beautiful single neuron activity. But we are interested in the human behavior. And in humans, we cannot record single neurons. So we have to go more towards measurements like mobile EEG. And here, though, the problem is that the signal to noise ratio is way, way, way lower. So we have way more or way less signal gain already compared to single neurons. And what I am currently working on is to establish, or what we have done already is to have a mobile EEG system in-house. Currently, we are working on establishing the room, and then we can also have mobile EEG studies with some mobile eye tracking and video-based motion tracking. And this is a very recent result here. If you want to see that, you can see here me going around. I will go out to take the calibration board. And here's the 3D reconstruction based on computer vision-based methods. These black dots are cameras that we have. We have 5 in total. One is on the top and then each of the walls. We can get the 2D key points. We can triangulate it based off this key calibration board. And then we get this 3D full skeleton model. So also that part is working. We have to synchronize views across cameras. And the task for this, the first task for this will be that they again walk through the room. They will receive now auditory cues and they will have to evaluate whether they have to go left or right towards the end of the room. And then they have to move until the end and then turn to the corresponding corner. And here now the question for us is, can we extract now decision signals during movements in human adults and children using mobile EEG? That has just not been shown so far and we just don't know. And that is also what leads me to the current challenges and projects. So one part would be the methods advancement. And this is now also like giving you some hints towards if you decide to write a proposal, what you could focus on. But of course, I'm very happy about any other project proposals or ideas. So in terms of methods advancement, it would be, of course, we have to figure out now pre-processing of mobile EEG in freely moving adults, and most importantly, of pipelines that would work in children because they just move differently. They have different type of movement artifacts. And to also train and apply video-based motion tracking on kids from 6 to 12, because the model I've been showing you is like many other models out there trained on human adults. So it predicts very well when it's adults. But when you go smaller, it does not necessarily scale. And it does especially not scale to all type of movements that the kids have. So very rapid movements sometimes, very fast movements. And we need to train a model and adapt to that. Scientifically, and I think that's probably the more interesting part for you, is when do we know now what was the decision time point? Because now we're instructing the participants to go until the end of the room and then turn. Can we extract somehow where the decision time point was by, for example, analyzing the velocity or the velocity of or the gait speed or the steps? Like, is there some type of markers in the behavior still that would indicate now we're deliberating versus now we're decided and now we're committing? And then the question, of course, would be for us, given that also you guys are neural data scientists, is the neural correlates of decisions during walking. So to model the data such that we can be quite confident it is the decision, it is a commitment to go to right or left that is driving the neural activity. It's not pose change, it's not head gaze shift, it's not motion artifacts. So basically trying to reproduce something that we've done in monkeys now in humans. I would like to jump back to monkeys again. So before I showed you there was a task, it was in a freely moving environment. That was super cool, super exciting already. It has not been shown before. But real life is not repeated trials necessarily. I mean, sometimes we have some tasks that we do that are repeated and always the same. But if I'm going through the city center, if I'm going for a hike, it's not all repeated same patterns of movements, but it's more free movements. So what we want to go to next is how does decision signals look like during free movements in macaques? So you don't have trials anymore, so you have continuous behaviors, and now you have to extract the decision time points. And here the task of the monkey was basically they had these different stations. So here are two screens where they can receive some fluid rewards. Then here are some wood patches, as you can see here. Then there are fruits or vegetables that they like attached to it. And then we have these four hanging branches where there were always grape hanging up there. And the monkeys had to go there, stand on two legs, pull it down to get the grapes. And we can get very nice tracking again. So this is an example that I want to show you. So here's the real video again, and here's our monkey taking those hanging grapes. Now you can also see them. So that part is there, and we can see that. That's fantastic. But the current challenge or also potential projects could be if you would decide to think about this in this line, and if I manage to excite you about this, how to extract decision time points from these continuous free behaviors. How do we know at which time were they not just walking, for example, from here towards the screen without having the intention to go there versus they wanted to go there? And to just explain this a bit more, I want to show you this idea here. So we do think that often we are goal directed. We wanted to go to position A or B, and we are goal directed. We just go there directly. Sometimes we are just going, we're just strolling around, and then on the way we decide, now actually I want to go to this shop or I want to go to that shop. And that's exactly the two examples. So here it seems you wanted to turn here, but then now and now I do a small stop here. So it's a bit like roaming and strolling. Whereas here it becomes very evident since the beginning. He wants to go here. He looks there, he goes there. So that leads to the different type of challenge, which is how can we distinguish between free roaming and strolling versus goal-directed behaviors? So how would the model look like, and which behavior parameters would be informative for this distinction? And once we should manage this, then of course we're super interested in looking at the neural activity and see how does it differentiate between goal-directed versus roaming, strolling walks. And one thing that I skipped before because I wanted to make this point is another very, very urgent question that we have right now is trying to come up with a model that can help us to determine what determines the next goal. So if I am here, what is the environmental factor that makes it happen that then the monkey says, now I want to go exactly to this patch and not to this one, for example? Is it the body posture? Is it the proximity? Is it the head gaze? Is it where I have been before already? Etc., etc., etc. So that would be really cool if we can try to address this question and try to model it a bit. Now, this part I've talked about. Sorry for the back and forth. That already leads me to the end. So thank you very much for going through this, listening to me. One of the upcoming project or immediate upcoming project is also we want to transfer that mobile EEG study that you've seen with humans into the VR environment. And for that, we are really also actively looking for people who would be interested in that or um, want to translate the paradigms into VR and also program VR. Independent of that, I am currently really actively looking for interns, bachelor's, and also master thesis students. So if in case I can get you excited for some of these projects or you have any other ideas or any other suggestions, please just contact me. I would be very happy to hear from you. Also, if you have any questions, just hit me an email. And with this, I thank you very much and wish you a wonderful new week.